Welcome back. In today's class, we will be learning about Proteobacteria. Proteobacteria, in few of the books, is also referred as Pseudomonas. So, in Proteobacteria, they come under atypical prokaryotes. When we learn about typical prokaryotes, they have significant characteristics, but then in atypical prokaryotes, they have a few exceptional organisms under them. They might show complex reproduction, they might show varied metabolism, or they might show varied motility, or utilization of any unusual element. All these are classified under atypical prokaryotes. One such are Proteobacteria. They are simply classified as alpha, beta, gamma, delta, and epsilon. We will be learning their general characteristics with a small example under each. Proteobacteria almost has about 2000 species under them. They all are believed to be gram-negative in nature. They are obligate intracellular parasite. They can also be free-living. They are chemolithotrophic or autotrophic, or uh, uh, chemoheterotrophic in nature. They might have few specialized structures such as prostaceae or stalks, buds, sheaths, or fruiting bacteria shown by them. These are not generally put forth by any typical prokaryote. They are dependent in nature. They can also be heterotrophic, or they can also be autotrophic in nature. They classify all the bacteria, most of the bacteria under them. They have about 2000 species as mentioned. They show varied motility. Few are anaerobic in nature, few are facultative anaerobes, or obligate anaerobes are also there. The name of the phylum is named after the Greek word, the god of seas, Proteus. That is how we regard God capable of assuming different shapes. That is how this Proteo or Proteus came into the picture, which shows indefinite characters, which shows a non-systematic modifications or evolutions. That is why they call it as god of seas, which include lot of organisms under Proteobacteria. Proteobacteria itself is a very vast term which includes lot of organisms under it, showing varied characteristic organisms. It is classified in the Burgess Manual in Volume 2, which speaks about all the types of Proteobacteria. All the classifications are seen. They can starting from their morphology to their genotypical or phenotypical characteristics are well quoted in Burgess Manual Volume 2. 16S rRNA sequencing, yes, is to identify the species level. So to speak about Proteobacteria and its classification, they have alpha Proteobacteria, which includes nitrogen fixing organism, that is Rhizobium agrobacterium. Beta Proteobacteria includes many of the sulfur or ammonia oxidizing organisms. Gamma Proteobacteria is about Pseudomonas or Xanthomonas or any acid-loving organisms. Delta Proteobacteria is iron or sulfur-reducing organisms. Epsilon Proteobacteria is about symbiotic or pathogenic organisms, and Zeta Proteobacteria is the sole member for Mariproteus organism. This is the classification according to the Burgess Manual, where organisms are classified under each. There are numerous organisms. We will be learning one under each or type. So, alpha Proteobacteria, beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, and epsilon Proteobacteria is what we will be learning in today's session. So to come under alpha Proteobacteria, we have purple non-sulfur bacteria is Rhodospirillum or Azospirillum. These are non-sulfur bacteria which cannot store sulfur as in its granule. Then you have Rickettsia, and you have nitrogen fixing bacteria like your Rhizobium. In alpha proteobacteria, they are believed to be oligotrophic in nature, those capable of living or surviving even under low nutritional level. They are methylotrophic in nature, like your methylobacteria. They have unusual metabolism that they can oxidize methane. And there are few that are capable of fixing nitrogen, like your nitrobacter or your rhizobium. They are capable of involved in nitrogen fixing activity. About 7 orders or 20 families are there under alpha proteobacteria. These are the classification or phylogenetic relationship shown by your alpha proteobacteria. To speak about purple non-sulfur bacteria, we have rhodospirillum or azospirillum. These are the non-sulfur bacteria, comes under alpha proteobacteria, photoorganoheterotrophic in nature. They are or can also be chemoorganoheterotrophic in nature. They behave in the absence of light. They do not metabolize sulfur. They live in very nutrient low or nutrient deprived environment. Oxygen inhibits bacteriochlorophyll and carotenoid synthesis, so that cultures are growing aerobically in the dark are colorless. So next, speaking about rickettsia, we all know rickettsia, is a pathogen. Rickettsia rickettsii is a pathogen which creates the disease rickettsia. Rickettsial infection is generally seen in the epithelial cell, induces, production of more and more rickettsia subunits inside. Because of the presence of the protein, the tail protein, actin protein, they are capable of showing certain type of motility, which makes them more pathogenic in nature, reproduce well, rupture from the host cell and affect the new healthy cell. All these are seen by your rickettsial cell cycle, which is parasitic or mutualistic in nature, capable of showing motility with the help of the actin protein tail. Nitrifying bacteria again are chemolithotrophic in nature. They are capable of fixing nitrogen. They work in aerobic condition, gram negative in nature, rod-shaped bacteria. They can also be spherical or lobate structure. There are few nitrosomonas or nitrospira which is in gamma proteobacteria. So under alpha proteobacteria, one nitrogen fixing bacteria is your Nitrobacter, which is capable of fixing nitrogen in leguminous plants. So this nitrification process or your nitrogen cycle is been highly influenced by Nitrobacter. So these are the this is a consolidated table of alpha proteobacteria, which has numerous organisms under it. Its morphology, its a G+C content, genome structure, oxygen requirement, and its distinctive characteristics. Then comes the beta proteobacteria. These are the organisms that have additional capability to oxidize hydrogen, ammonia, methane, volatile fatty acids, and similar substances. These have about 7 orders and 12 families are under it. So these are the phylogenetic relationship of the beta proteobacteria. So under hydrogenophilales, we have thiobacillus, like nitrifying bacteria, and colorless sulfur bacteria, which is unicellular in nature, rod-shaped bacteria or spiral-shaped bacteria, sulfur oxidizing bacteria. They could be motile or non-motile. Under Neisseria, we have Neisseria organism, which causes Neisseria gonorrhea, which causes gonorrhea disease. That comes under Neisseriales, which is non-motile, aerobic, gram-negative cocci. They are capsulated. They have fimbriae. They are chemoorganotrophic in nature. They can also be catalase and oxidase positive. Nitrosomonadeals, they are chemolithotrophic in nature. They also contain nitrifying bacteria like your nitrosomonas. So these are your sulfur oxidizing organisms. As I told, the significant characteristic of a beta proteobacteria is to oxidize sulfur or hydrogen or any particular type of oxidizing capability is seen in your beta proteobacteria. This is a consolidated table of about their nutritional type, sulfur deposition characteristic, its structure, G plus C content. Then comes the gamma proteobacteria. They are chemoorganotrophic in nature or facultative anaerobic. Here they are also believed to be photolithotrophic or methylotrophic in nature. These are the organisms that comes under gamma proteobacteria, which includes some known organisms like your Pseudomonas or Photobacterium or Vibrio cholerae or your Proteus or your E. coli or Salmonella, Citrobacter. All this comes under gamma proteobacteria. These includes purple sulfur bacteria. Purple non-sulfur bacteria is an alpha proteobacteria. Purple sulfur bacteria is in gamma proteobacteria, which has a capability to oxidize hydrogen sulfide to sulfur and deposit it in the internal cavity of the cell as sulfur granules. They are strict anaerobes in nature. They are photolithotrophic in nature. Next comes the delta proteobacteria. This is a phylogenetic relationship of your delta proteobacteria, which includes Bividovirus and Myxobacteria. Myxobacteria is also involves a gliding bacteria, which shows a typical type of gliding motility. So under your gamma delta proteobacteria, we have various types of organisms, their characteristic morphology, G plus C content, oxygen requirement, and distinctive characteristics, under which Myxobacteria plays a vital role. So for under delta proteobacteria, Myxobacteria is the best example. So Myxobacteria is also called as, gliding bacteria, which shows a distinctive type of motility and also distinctive type of reproduction. In reproduction, it produces something called as fruiting bodies. Inside fruiting bodies, myxospores will be present. When myxospores escape from the fruiting bodies, falls on a suitable substratum, under the influence of a right environmental condition, these germinate into a new vegetative cells. So this shows also a typical, not only a atypical type of motility, but also a typical type of reproduction. They are generally seen in soil, gram-negative in nature, aerobic in nature, shows gliding motility, complex life cycle by producing fruiting bodies, and myxospores are produced, which are believed to be dominant in nature or dormant in nature. It has a high G plus C content, about 71%. Here, Myxococcales is divided into 6 families, and based on the shape of their vegetative cell or their sporangium or their myxospore. Sporangium is nothing but again the fruiting body. Life cycle is complex, reproduction is complex as it. So this is the reproduction. This is how the myxobacterium looks like. This is how the myxobacterium looks like, and myxospores are produced, aggregate of myxobacterium, and fruiting body will be produced, inside which numerous myxospores will be present. So this is how it looks. This is the ultra structure of your fruiting body, which has a stalk and numerous sporangium attached to it. Inside the sporangiole, there is myxospores. So the last type of proteobacteria is epsilon proteobacteria, which is slender, gram-negative, rod-shaped. It can also be helical or vibrioid. It includes 2 important organisms, Campylobacter and Helicobacter. This is moderately seen in relatively high temperature. They are thermophilic in nature. They could also be chemolithotrophic in nature or chemolithoautotrophic in nature. They are strict anaerobes. They have the capability to oxidize hydrogen and use sulfur as an electron acceptor. They can either be free living or symbiotic in nature, but a significant characteristic of your epsilon proteobacteria is its ability to survive harsh environmental condition, that is high temperature. Epsilon proteobacteria, the main organisms are Campylobacter and Helicobacter. These are believed to be microaerophilic in nature, highly motile, helical or vibroid shaped. Yes, of course, gram-negative, rod-shaped. They could be non-pathogenic, or few are pathogenic in nature, which causes reproductive diseases. Your Campylobacter is which causes reproductive diseases, especially in mammals. So Helicobacter again causes certain stomach or upper respiratory tract or intestinal diseases. It causes ulceration in humans also. It feeds on the internal lining of the intestinal membrane and creates a gastritis or stomach ulcers in mammals or even in human beings. So that is all we have for proteobacteria. So from where did we start? We started from the introduction. What exactly are proteobacteria? What are its general characteristics? How is it scheduled in the Burgess manual? The examples of each type of proteobacteria. So starting from alpha proteobacteria, we learned about purple non-sulfur bacteria, rickettsia, and nitrifying bacteria. And then beta proteobacteria, we have their classifications and sulfur oxidizing capability. Gamma proteobacteria, we learned about purple sulfur bacteria. Delta proteobacteria, we learned about the capability of your myxobacterium to show varied motility and reproduction with the diagram. Then comes the epsilon proteobacteria, capability to survive in harsh environmental condition. The best example would be your Campylobacter and your Helicobacter. So thank you so much. Have a good day.